Welcome to Softly Curious. Tonight, we begin with something small enough to rest quietly inside your skull and powerful enough to generate every memory you have ever kept. The human brain does not announce its strength. It does not glow or vibrate or make its effort visible. It works in silence, inside darkness, consuming energy, forming patterns, and maintaining a scale that is easy to underestimate. If you were to hold a human brain in your hands, it would weigh roughly 1.5 kilograms. That is about 2% of total body weight for an average adult. And yet, despite its modest mass, it consumes close to 20% of the body's total energy. In metabolic terms, it behaves less like a passive organ and more like a city that never sleeps. At rest, the brain uses roughly 20 watts of power. 20 watts is enough to light a small LED bulb. It does not sound impressive at first, but that steady output sustains thought, memory, language, emotion, balance, vision, and the ongoing regulation of the body. It is not bursts of brilliance that define the brain's power. It is constancy. Beneath that steady energy use lies a structure of remarkable scale. For many years, textbooks estimated that the human brain contained around 100 billion neurons. The number was repeated so often that it became a kind of scientific folklore. But in the early 21st century, Brazilian neuroscientist Susana Herculano Hausel applied a refined cell-counting method and arrived at a more precise estimate, approximately 86 billion neurons. 86 billion is still a number large enough to resist intuition. Each neuron is a living cell capable of generating electrical signals, extending branching dendrites to receive input, and a longer projection called an axon to send output onward through the network. These cells do not function in isolation. They form connections that transform individual signals into coordinated patterns. Every neuron can establish thousands of synapses, specialized junctions where chemical and electrical messages pass from one cell to another. And when billions of neurons are each capable of forming thousands of these connections, the total number of synapses reaches into the trillions. Current estimates suggest that the human brain contains on the order of 100 trillion synaptic connections, creating a network of staggering density where information is constantly being rooted, strengthened, or diminished. This network remains in motion at all times, with electrical impulses traveling along axons at speeds shaped by a fatty insulating layer called myelin, which increases conduction efficiency and allows signals in heavily myelinated fibers to reach speeds exceeding 100 meters per second. At that rate, information can traverse the length of the brain in mere milliseconds, enabling coordination between distant regions without noticeable delay. Such speed is rarely perceived consciously, yet without it fluent speech, balanced movement, and coherent perception would fragment into hesitation and confusion. The brain's rapid communication is not traumatic or visible, but it is foundational to everything that feels seamless in experience. The cerebral cortex, forming the outer layer of the brain, plays a central role in this computational capacity. If unfolded and spread flat, the cortex would cover roughly 2,000 square centimeters, an area comparable to a large dinner napkin, achieved through an intricate pattern of folds known as gyri and sulci that allow greater surface area to fit within the confined space of the skull. These folds are not decorative features, but spatial solutions, enabling a higher density of cortical neurons and expanding the brain's processing potential without increasing head size. Compared with our closest evolutionary relatives, humans possess not only a larger brain in absolute terms, typically weighing between 1,300 and 1,400 grams, but also a greater proportion devoted to the cerebral cortex. A chimpanzee brain weighs approximately 400 grams, and while the difference in size is significant, the more meaningful distinction lies in neuronal organization and density within regions associated with language, abstract reasoning, and long-term planning. This expansion of scale did not introduce obvious physical markers visible from the outside. There are no surface inscriptions on the skull that signal imagination or symbolic thought. The difference resides in cellular architecture and network complexity rather than outward form. Estimating the brain's storage capacity introduces further complexity because memory is not stored as discrete files but distributed across changing patterns of synaptic strength. Nevertheless, theoretical models suggest that if synaptic states are treated as units of information, the brain's potential storage could extend into the petabyte range, where a single petabyte equals 1 million gigabytes. Although such figures rely on assumptions about encoding efficiency, even conservative calculations indicate that the brain's capacity is vast and far exceeds typical conscious recall. Capacity, however, is not limited to storage alone. It also includes dynamic computation. At every moment, millions of neurons fire in coordinated patterns, producing structured and rhythmic activity rather than random noise. Even in periods of quiet wakefulness, when a person lies still with eyes closed, networks such as the default mode network increase activity, suggesting that the brain reorganizes rather than shuts down. Unlike skeletal muscles, which increase energy consumption proportionally with effort, the brain maintains a relatively stable total energy expenditure across different mental tasks. When solving a complex problem, it does not dramatically increase its overall power usage, but instead redistributes activity across specialized regions, reflecting an efficiency that favors balance over spikes in demand. This efficiency is one of its defining characteristics, subtle yet continuous. Deep within the temporal lobe lies the hippocampus, a structure central to spatial navigation and memory formation. In a well-known study of London taxi drivers who completed the demanding memorization process, known as the knowledge, researchers observed increased gray matter volume in the posterior hippocampus compared to individuals without such training. Gray matter, meaning regions rich in neuronal cell bodies, appeared to reflect the long-term cognitive demands placed on these drivers. The findings did not suggest limitless expansion, but demonstrated that sustained use correlates with measurable structural differences detectable through imaging technologies, such as magnetic resonance imaging. The brain records repeated experience, not only in memory traces, but in subtle anatomical variation. Across regions, similar principles appear to apply. Prolonged engagement in specific cognitive activities can be associated with modest structural differences, changes too subtle to detect externally, but observable through careful measurement. Beneath the surface, the overall scale remains constant. 86 billion neurons interconnected through trillions of synapses. A folded cortex maximizing surface area, signals traveling at remarkable speeds along myelinated pathways, and a structure that consumes roughly 1/5 of the body's energy, while representing only a small fraction of its mass. Yet none of this feels heavy. Forming a sentence does not produce strain. Recalling a name does not create visible exertion, and the orchestration of movement unfolds without conscious calculation. The brain's power is experienced not as effort, but as continuity, a steady background process that sustains thought without announcing its magnitude. Each thought emerges from coordinated patterns shaped over years. Each memory rests upon stabilized synaptic pathways, and each perception depends on signals processed in fractions of a second. The numerical scale provides perspective, but the deeper realization comes from understanding that this scale operates continuously and without spectacle. The brain does not pause between tasks, nor does it fully power down at night. Even during sleep, electrical rhythms persist, consolidating information and maintaining internal balance. Its strength is not explosive or episodic, but persistent and measured, residing in density, organization, and efficiency. If its activity were visible, it might resemble a vast constellation of signals moving across a folded landscape enclosed within bone. From the outside, however, there is only stillness, while inside that stillness billions of cells remain in coordinated conversation, sustaining an inner world with quiet, unbroken power. The strength of the brain is not only measured in numbers or electrical speed, but in its ability to change itself. This capacity is known as neuroplasticity, a term used to describe the brain's ability to reorganize its structure and function in response to experience. Neuroplasticity does not imply limitless transformation. It refers to the measurable ways in which neural pathways adjust when exposed to repeated activity, injury, learning, or environmental change. For much of the 20th century, scientists believed that the adult brain was largely fixed, that after childhood, its wiring became stable and resistant to modification. That view began to shift as experimental evidence accumulated, demonstrating that even mature neural circuits retain the capacity to reorganize. The brain is not a static map. It is a system that edits itself. One of the clearest demonstrations of this adaptability appears in stroke recovery. When a stroke damages a specific region, such as the motor cortex responsible for voluntary movement, patients may initially lose control over certain muscles. Over time, and with targeted rehabilitation, neighboring or connected regions can begin to assume aspects of the lost function. This does not represent a perfect restoration, but it shows that surviving neural networks can reorganize, forming new pathways that compensate for damaged ones. Research led by neuroscientist Michael Merzenich played a significant role in reshaping how plasticity was understood. Through experiments involving sensory remapping in animals, Merzenich demonstrated that when sensory input changes, cortical maps adjust accordingly. Areas of the brain devoted to processing specific inputs can expand, contract, or shift depending on usage patterns. The cortex is not rigidly assigned. It is responsive. In individuals who are blind from an early age, imaging studies reveal that the visual cortex, normally dedicated to processing visual information, does not remain inactive. Instead, it becomes involved in processing touch and sound, particularly during tasks such as reading Braille. This functional reassignment illustrates that cortical regions are defined not solely by location, but by input. When one channel closes, another can be amplified. Plasticity is also evident across the lifespan, though its intensity varies. In childhood, the brain exhibits heightened flexibility, allowing rapid acquisition of language and sensory motor skills. Neural circuits form quickly, shaped by exposure and repetition. In adulthood, plasticity remains present, but operates within narrower constraints, often requiring sustained practice and focused attention to produce measurable change. The difference is not absence versus presence, but degree. Studies comparing bilingual individuals to monolingual speakers have identified structural differences in regions associated with language control and executive function. Some research suggests increased gray matter density in areas responsible for managing multiple linguistic systems, reflecting the cognitive demand of switching between languages. These findings do not imply superiority, but they demonstrate that sustained mental practice can correlate with anatomical variation. Musicians offer another window into adaptive change. Long-term instrumental training has been associated with measurable differences in the motor cortex and in regions responsible for auditory processing. String players, for example, often show expanded cortical representation for the fingers of the left hand, which performs intricate movements on the instrument's neck. Repetition shapes representation, and representation reflects use. Adaptation is not limited to skill acquisition. Traumatic experiences can also leave structural and functional traces. Chronic stress has been associated with changes in regions, such as the amygdala and prefrontal cortex, affecting emotional regulation and attention. These alterations do not represent irreversible damage in every case, but they highlight that experience, whether constructive or harmful, interacts continuously with neural architecture. Underlying these shifts is a process known as synaptic pruning, meaning the selective elimination of weaker or unused connections. During development, the brain produces more synaptic connections than it ultimately retains. Through experience and activity, frequently used pathways are strengthened while others are reduced. Pruning increases efficiency, refining circuits so that signaling becomes more precise. Adaptation, therefore, is not simply growth. It is careful editing. The brain's ability to rewire itself does not announce transformation with visible signs. It unfolds quietly through altered pathways and adjusted sensitivities, reflecting the patterns of life that pass through it. Change is neither chaotic nor limitless, but structured and responsive, shaped by repetition, recovery, language, music, stress, and learning. In this way, flexibility becomes another form of strength, less about magnitude and more about resilience. There are moments when the human brain appears to step slightly beyond the ordinary range of performance, not in spectacle and not in myth, but in carefully documented cases that reveal unusual configurations of memory, perception, and calculation. These cases do not represent a different species of mind. They are variations within the same architecture, showing what can happen when certain circuits become highly specialized. One of the most studied examples is savant syndrome, a condition in which individuals with developmental differences demonstrate extraordinary skill in a narrow domain such as music, mathematics, calendar calculation, or visual memory. Savant abilities are rare, and they often coexist with significant cognitive challenges in other areas. What makes them scientifically compelling is not mystery, but specificity. The skills are measurable, repeatable, and often precise beyond conventional training. A widely known case is Kin Peek, who served as partial inspiration for the film character in Rain Man. Peek possessed an exceptional memory, reportedly able to recall the contents of thousands of books after reading them once. He did not simply memorize passages. He retained detailed factual information across history, geography, and literature. Neuroimaging later revealed structural differences in his brain, including atypical connectivity between hemispheres. His abilities were not magical. They were neurological. Another documented case is Daniel Tammet, who combines numerical and linguistic talents with the ability to describe his internal experience. Tammet reports perceiving numbers as shapes, textures, or colors, a phenomenon related to synesthesia, meaning a blending of sensory experiences. He has recited thousands of digits of pi from memory and learned new languages in remarkably short periods. What distinguishes his case is not only performance, but introspection. He can articulate how his mind organizes information. These individuals demonstrate that the brain can, under certain developmental conditions, amplify particular abilities while leaving others less developed. Research into savant syndrome suggests that enhanced local connectivity or reduced inhibitory filtering may allow details to be processed with unusual intensity. Rather than adding new capacity, the brain may be redistributing emphasis. A different but equally intriguing ability is absolute pitch, also known as perfect pitch, defined as the capacity to identify or produce a musical note without any external reference tone. Most trained musicians rely on relative pitch, comparing one note to another. Individuals with absolute pitch can hear a single tone and label it immediately, such as identifying a note as A flat or C sharp. Studies suggest that early musical exposure during a critical developmental window increases the likelihood of developing this skill, and imaging research indicates subtle structural differences in auditory regions. Absolute pitch does not require extraordinary intelligence. It reflects precise auditory categorization. Memory itself can, in rare cases, operate with unusual persistence. Jill Price is one of the first individuals formally documented with hyperthymesia, a condition characterized by highly superior autobiographical memory. She can recall specific dates from decades earlier and describe events from those days with striking detail. When asked about a random date, she does not compute the answer. She remembers it, Brain imaging studies of individuals with hyperthymesia have revealed differences in regions associated with autobiographical recall. Although the condition remains poorly understood, it is not flawless memory in all domains. It is specific to personal experience. Beyond rare named cases, the brain also possesses systems that temporarily enhance performance under threat. The body's response to danger involves the release of adrenaline, a hormone that prepares the organism for rapid action. In such moments, perception can shift. Many people report that time seems to slow during accidents or sudden emergencies. Research suggests that this altered time perception may result from heightened attention and increased density of memory encoding, rather than an actual change in temporal flow. The brain records more information in a short interval, which later creates the impression of expanded time. Pain perception offers another example of modulation. The brain contains descending inhibitory pathways that can suppress pain signals at the spinal level. In life-threatening situations, individuals may sustain injuries without immediately feeling pain. This phenomenon is not psychological denial. It is neurochemical regulation. Endorphins, meaning endogenous morphine-like substances produced within the body, bind to opioid receptors and reduce pain transmission. The suppression is temporary, but it illustrates that perception is actively managed. Competitive memory athletes provide a different perspective on exceptional ability. Participants in international memory championships perform tasks such as memorizing long sequences of numbers, shuffled decks of cards, or random words within limited time frames. Neuroimaging studies have shown that these individuals do not necessarily have larger memory regions. Instead, they employ structured techniques such as the method of loci, a strategy that associates information with imagined spatial environments. Their performance reflects disciplined training rather than innate anomaly. The brain responds to strategy. These examples share a common feature. They do not represent supernatural enhancements. They reflect variations in attention, connectivity, inhibition, and practice. In some cases, structural differences are present. In others, deliberate training shapes outcome. The brain appears capable of focusing intensely on specific domains, sometimes at the expense of balance, sometimes through sustained repetition. It is important to note that exceptional performance often coexists with vulnerability. Savant abilities may be accompanied by social or communicative challenges. Hyperthymesia can be psychologically burdensome, as unwanted memories remain vivid and intrusive. Absolute pitch, while musically advantageous, can cause discomfort when music is slightly out of tune. These abilities are not universally beneficial. They are specialized. When observing rare minds, the temptation is to ask how much more the brain might do if fully unlocked. Scientific evidence suggests a more measured conclusion. The brain does not hide vast unused reservoirs waiting for activation. Instead, it operates within biological constraints while allowing certain pathways to strengthen dramatically under particular conditions. Extreme ability does not require a different organ. It arises from the same cellular foundation present in every human brain. The difference lies in distribution, development, and experience. Certain circuits become highly refined, whether through developmental divergence, early exposure, or sustained practice. In quiet laboratory settings and in documented biographies, these cases continue to be studied with caution. Each offers insight into how flexible neural systems can become when shaped by unusual parameters. They do not overturn the rules of biology. They illuminate them. The exceptional mind does not break the structure of the brain. It reveals how selectively that structure can be tuned. Lucid dreaming is one of the most studied examples. A lucid dream occurs when a person becomes aware that they are dreaming while still inside the dream. This awareness can range from a faint recognition to a stable sense of reflective control. The dream does not disappear. Instead, The dreamer observes it from within. Research into lucid dreaming gained scientific traction through the work of Stephen LeBerg, who demonstrated in controlled sleep laboratory settings that lucid dreamers could signal their awareness using predetermined eye movements. Because eye muscles remain active during REM sleep, meaning rapid eye movement sleep, these signals could be detected on electroculogram recordings. The experiments provided objective markers of subjective awareness. REM sleep itself is a paradoxical state. Brain activity during REM resembles wakefulness in many respects, with vivid imagery and emotional intensity. However, during typical REM episodes, the prefrontal cortex, particularly regions involved in self-monitoring and logical evaluation, shows reduced activity. In lucid dreaming, neuroimaging studies suggest partial reactivation of prefrontal areas. Awareness appears to return without fully dissolving the dream environment. This combination of activation and inhibition reveals that consciousness is not an all-or-nothing condition. It is layered. The brain can generate immersive internal worlds while selectively adjusting reflective oversight. Lucidity does not create new structures. It modifies balance. A different but equally structured state is known as flow, a term popularized by psychologist Mihaly Csikszentmihalyi to describe periods of deep absorption in a task. During flow, attention narrows, self-consciousness diminishes, and the perception of time often shifts. Athletes, musicians, surgeons, and writers have all reported entering this condition. Neuroimaging studies suggest transient hypofrontality, meaning reduced activity in certain self-referential prefrontal regions, allowing procedural and motor networks to operate with minimal interference. Meditation offers another window into altered awareness. Long-term practitioners of focused attention or open monitoring meditation display measurable changes in brain activity patterns. Electroencephalography, abbreviated as EEG, meaning the recording of electrical activity along the scalp, has revealed increased gamma wave activity in experienced meditators during certain practices. Gamma waves are high-frequency oscillations associated with integrative processing across distributed networks. In studies involving Tibetan Buddhist monks with thousands of hours of meditation experience, researchers observed sustained gamma synchrony during compassion-focused meditation. These measurements were not anecdotal reports. They were recorded through electrodes, analyzed statistically, and replicated across sessions. The findings suggest that deliberate mental training can shape not only attention, but measurable neural rhythms. The placebo effect further illustrates the brain's capacity to alter perception through expectation. When individuals receive a treatment they believe to be active, even if it is inert, measurable physiological changes can occur. Neuroimaging has shown activation in pain-modulating regions such as the anterior cingulate, cortex, and the periaqueductal gray during placebo analgesia. Expectation triggers endogenous opioid release, altering subjective experience through identifiable pathways. This demonstrates that belief is not a vague psychological overlay. It engages defined neural circuits capable of influencing sensory processing. The brain anticipates outcomes and adjusts perception accordingly. Expectation becomes biology. Dissociative states provide a more complex example of altered awareness. Dissociation refers to a disruption in the normal integration of memory, identity, perception, or consciousness. In mild forms, it may manifest as daydreaming or feeling temporarily detached from surroundings. In more severe cases, it can involve fragmented memory or altered identity states. Neuroimaging studies suggest changes in connectivity between frontal regulatory regions and limbic structures during dissociative episodes. Across lucid dreams, flow, meditation, placebo responses, and dissociative states, a common principle appears. The brain does not exit reality. It reconfigures internal coordination. Certain networks amplify while others quiet. Awareness shifts not because structure collapses, but because emphasis changes. These altered states reveal that consciousness is flexible within biological limits. The same organ that maintains stable waking perception can generate immersive dreams, focused absorption, analgesic belief responses, and temporary detachment. None of these require supernatural explanations. They emerge from modulations in timing, connectivity, and inhibition. The brain's capacity for altered awareness is not an exception to its function. It is part of its design. Deep within the center of the brain, almost perfectly positioned between the two hemispheres, lies a small structure no larger than a grain of rice. It sits in a narrow groove where the two halves of the thalamus join, tucked safely beneath the cerebral cortex and behind the third ventricle. The pineal gland is part of the endocrine system, meaning it releases hormones directly into the bloodstream, rather than sending electrical signals like neurons do. Its primary product is melatonin, a hormone that helps regulate the body's internal timing. Melatonin is often described as the sleep hormone, but its role is broader than inducing drowsiness. It functions as a signal of darkness. Unlike many other glands, the pineal does not operate independently of the external world. It is tightly linked to light. The process begins in the retina, the light-sensitive layer at the back of the eye. Specialized cells in the retina detect environmental light levels and transmit this information along a neural pathway that ultimately influences the pineal gland. When light reaches the eyes, signals travel through the optic nerve to a small region in the hypothalamus called the suprachiasmatic nucleus. The suprachiasmatic nucleus, often abbreviated as SCN, acts as the brain's primary circadian clock, meaning it coordinates biological rhythms that follow an approximately 24-hour cycle. The SCN receives direct input from the retina and adjusts its timing based on external light cues. From there, it sends signals through a multi-step pathway that either suppresses or permits melatonin production in the pineal gland. When daylight is present, the SCN inhibits melatonin release. As evening approaches and light diminishes, inhibition decreases, and the pineal begins secreting melatonin into the bloodstream. Levels typically rise in the evening, peak during the night, and fall again toward morning. The hormone does not force sleep, but it signals to tissues throughout the body that night has arrived. Circadian rhythm refers to this roughly 24-hour biological cycle that regulates not only sleep and wakefulness, but also body temperature, hormone release, digestion, and cognitive alertness. The term comes from the Latin circa diem, meaning about a day. These rhythms are endogenous, meaning they are generated internally, but they remain synchronized to environmental light through the retina-SC and pineal pathway. Even in complete darkness, the body would continue to follow a near 24-hour rhythm, though it would gradually drift without light cues. This phenomenon has been observed in controlled laboratory settings where participants are isolated from natural daylight. Over time, their sleep-wake cycles begin to shift slightly later each day, demonstrating that the internal clock is self-sustaining but not perfectly precise without environmental alignment. The pineal gland's melatonin output serves as one of the body's most reliable indicators of internal night. Because melatonin secretion follows a predictable curve, researchers often measure its concentration in saliva or blood to assess circadian timing. In sleep laboratories, the onset of melatonin release under dim light conditions is considered a key marker of biological evening. When this rhythm is disrupted, the effects become noticeable. Jet lag provides a clear example. Rapid travel across time zones creates a mismatch between the internal circadian clock and the external light-dark cycle. The SCN continues to operate according to the original time zone, while environmental light cues signal a new schedule. Until alignment gradually occurs, individuals may experience insomnia, daytime fatigue, digestive changes, and impaired concentration. The pineal gland adjusts slowly, shifting melatonin timing by small increments each day. Shift work can create a similar disruption. Individuals who work overnight under artificial lighting and attempt to sleep during the day often struggle because melatonin production remains linked to light exposure. Bright light at night suppresses melatonin secretion, making it difficult to initiate restorative sleep afterward. Over extended periods, chronic circadian misalignment has been associated with increased risks for metabolic and cardiovascular conditions. Light itself is not uniform in its biological impact. Short-wavelength blue light, commonly emitted by LED screens and energy-efficient bulbs, is particularly effective at suppressing melatonin. Specialized retinal cells containing the photopigment melanopsin are especially sensitive to blue wavelengths. When these cells detect blue light, they send strong signals to the SCN, reinforcing the message that it is daytime. Evening exposure to bright screens can therefore delay melatonin onset, shifting sleep timing later than intended. The pineal gland does not measure time consciously. It responds to a chain of signals that begin with photons striking retinal cells. Through this pathway, environmental light is translated into hormonal rhythm. Darkness allows melatonin to rise, signaling the body to lower core temperature, reduce alertness, and prepare for sleep. Light reverses this process, suppressing melatonin and promoting wakefulness. The gland itself is composed primarily of pinealocytes, the cells responsible for melatonin synthesis. These cells convert the neurotransmitter serotonin into melatonin through enzymatic steps that become more active in darkness. Because serotonin levels fluctuate across the day, the pineal indirectly links mood-related neurochemistry to circadian regulation. The transformation from serotonin to melatonin is one of the body's quiet nightly transitions. Although small, the pineal gland is richly supplied with blood vessels. This vascularization allows melatonin to enter the bloodstream efficiently and circulate to distant organs. Cells throughout the body contain melatonin receptors, meaning the hormone signal extends far beyond the brain. The immune system, cardiovascular system, and reproductive system all respond in subtle ways to circadian timing. Importantly, melatonin does not function as a sedative in the traditional sense. It does not impose unconsciousness. Instead, it adjusts the timing of sleep by shifting the internal clock. Low doses taken at appropriate times can help realign circadian rhythm, particularly in cases of delayed sleep phase disorder or jet lag. However, timing is critical. Taken at the wrong hour, melatonin can shift rhythms in unintended directions. The pineal gland's activity also changes across the lifespan. In early childhood, melatonin production is relatively high, contributing to longer sleep durations. During adolescence, the timing of melatonin release shifts later in the evening, a biological factor contributing to the common pattern of teenagers preferring later bedtimes. In older adulthood, melatonin levels may decrease, sometimes correlating with fragmented sleep. Despite its size, the pineal gland serves as a biochemical bridge between the external environment and internal physiology. It translates light into timing, darkness into hormonal release, and daily cycles into coordinated bodily rhythms. Its function is steady and rhythmic, neither dramatic nor forceful, yet essential to the coherence of sleep and wakefulness. Each evening, without conscious command, melatonin levels begin to rise. Core body temperature slowly falls. Alertness softens. The shift is gradual and often unnoticed, yet it follows a pathway that begins with light entering the eye and ends with molecular changes in cells throughout the body. In this way, the pineal gland does not create time. It aligns the body with it. Long before microscopes and hormone assays defined its function, the pineal gland occupied a different space in human thought. Hidden deep within the center of the brain, and singular rather than paired, it appeared anatomically unusual. Most brain structures are mirrored across hemispheres. The pineal is not. That symmetry break, small but visually striking in dissection, invited interpretation long before measurement. In the 17th century, the French philosopher René Descartes proposed that the pineal gland might serve as the seat of the soul. Descartes was attempting to solve a philosophical problem: how an immaterial mind could interact with a material body. Because the pineal gland was centrally located and unpaired, he believed it might function as a point of integration between thought and physical action. His conclusion was speculative, rooted in anatomical observation, but not experimental evidence. At the time, neuroendocrinology did not yet exist. Descartes' proposal did not arise from mysticism, but from philosophical reasoning shaped by the scientific limits of his era. Without knowledge of neurotransmitters or circadian regulation, the pineal's isolation within the brain suggested importance. The idea that a single structure could coordinate perception and movement offered conceptual clarity in a period when physiology remained largely mysterious. Beyond Europe, symbolic interpretations emerged independently. In ancient Egyptian iconography, certain motifs resemble an eye placed centrally on the forehead, often referred to in modern language as the third eye. While historians caution against direct anatomical correlations, the symbolic association between inner vision and a central point within the head became culturally persistent. The metaphor of an inner eye conveyed awareness beyond ordinary sight. In several Eastern philosophical traditions, particularly within certain strands of Hindu and Buddhist thought, the concept of an inner eye, or Ajna chakra, is described as a center of perception beyond sensory input. Although classical texts did not identify this center anatomically with the pineal gland in a biological sense, later interpretations during the 19th and 20th centuries began linking the two. This association developed more through comparative symbolism than through empirical study. As modern anatomy advanced, scientific understanding of the pineal shifted toward measurable function. Yet symbolic interpretations did not disappear. Instead, they adapted. In contemporary alternative health discourse, claims sometimes emerge suggesting that the pineal gland becomes calcified and that this calcification suppresses intuition or consciousness. It is true that calcification of the pineal gland can occur with age, detectable through imaging as small mineral deposits. However, such calcification is common and not clearly associated with cognitive suppression. Some modern narratives attribute pineal calcification to fluoride exposure, proposing that fluoride accumulates in the gland and disrupts function. While fluoride can accumulate in calcified tissues, major health organizations and peer-reviewed studies have not established a causal link between typical fluoride exposure and loss of pineal endocrine function. The scientific literature remains cautious and does not support claims of widespread neurological impairment through this pathway. The persistence of these claims illustrates a broader pattern. When a structure occupies a hidden and central position within the brain, and when its function relates to sleep and cycles of consciousness, it becomes an inviting canvas for projection. Biological complexity often leaves conceptual gaps. Symbolic systems move quickly to fill them. Scientific inquiry, by contrast, advances through controlled measurement and reproducibility. Neuroimaging, hormonal assays, and anatomical studies define boundaries around what can be confidently stated. Beyond those boundaries lies uncertainty, not necessarily falsity, but untested possibility. The distinction matters. Science limits itself to claims that can be examined under defined conditions. Spiritual narratives and neuroscientific explanations operate under different frameworks. Spiritual systems often interpret inner experience symbolically, emphasizing meaning and transformation. Neuroscience seeks mechanisms, pathways, and measurable correlations. These approaches are not inherently hostile to one another, but they answer different kinds of questions. One asks what an experience signifies. The other asks how it occurs. The pineal gland, because of its modest size and central location, became a meeting point for both modes of interpretation. Historically, it served as a philosophical placeholder for the interface between mind and body. Culturally, it became a symbol of insight. Scientifically, it is understood as an endocrine organ regulating biological rhythms. Each layer reflects the knowledge and priorities of its time. What remains consistent is caution. Where evidence is strong, conclusions can be drawn. Where evidence is limited, restraint is appropriate. The pineal gland does not require mystification to be meaningful. Its documented role in regulating daily rhythm is already sufficient to anchor it in the architecture of human life. In the quiet boundary between history and measurement, the pineal reminds us that interpretation evolves. What was once described as the seat of the soul is now mapped through biochemical pathways. The questions have changed shape, but the center remains the same, a small structure, interpreted through the lens of each era's understanding. There is a quiet delay between what the brain begins and what we become aware of. We experience decisions as deliberate events. A thought appears, we weigh it, and then we act. Yet beneath that smooth sequence lies a layer of activity that unfolds before conscious recognition. The sensation of control may not be the starting point. It may be the narration that follows. In the 1980s, neuroscientist Benjamin Libet conducted a series of experiments that continue to influence debates about free will. Participants were asked to perform a simple voluntary movement, such as flexing a wrist, whenever they felt the urge. At the same time, their brain activity was recorded using electroencephalography. Libet focused on a signal known as the readiness potential, meaning a measurable buildup of electrical activity in the motor cortex that occurs before movement. What he observed was subtle but significant. The readiness potential began several hundred milliseconds before participants reported the conscious intention to move. In other words, the brain showed preparatory activity before the person became aware of deciding. The conscious decision appeared to follow the neural initiation. The delay was small, measured in fractions of a second, yet its implications were large. If neural preparation precedes awareness, then conscious intention may not initiate action in the way we intuitively believe. It may instead confirm or veto processes already underway. Later interpretations refined Libet's findings, suggesting that consciousness may still exert a veto power, meaning it can inhibit an action before execution. But the sequence remains revealing. Activity first, awareness second. Subsequent experiments using more advanced imaging techniques have replicated similar timing patterns. Neural signals predictive of a choice can sometimes be detected seconds before a participant reports making that choice. These findings do not imply that behavior is predetermined in a rigid sense. They indicate that decision-making unfolds gradually within distributed neural systems, with awareness emerging partway through the process. Much of daily decision-making occurs outside reflective thought. When selecting a route while driving or responding to a familiar question, the brain draws on stored patterns and learned associations without conscious calculation. These processes are efficient precisely because they are not deliberated each time. Conscious reasoning would be too slow for routine life. Intuition, often described as a sudden sense of knowing without explicit reasoning, reflects this background processing. Studies of intuitive decision-making suggest that the brain integrates prior experience, subtle cues, and emotional signals to produce rapid judgments. The outcome appears as a feeling rather than an argument. The reasoning exists, but it is distributed and largely inaccessible to introspection. The face provides another example of rapid, unconscious processing. Within a fraction of a second, the brain identifies a face, categorizes it as familiar or unfamiliar, and begins interpreting emotional expression. Regions such as the fusiform face area specialize in facial recognition. This recognition occurs before deliberate thought. You do not decide to recognize a friend's face. Recognition happens and awareness follows. Micro-expressions further illustrate the speed of unconscious evaluation. These are brief facial movements that reveal emotion, sometimes lasting less than a quarter of a second. Observers trained to detect them can identify concealed feelings that escape ordinary notice. Even without training, the brain registers subtle changes in expression and adjusts social responses accordingly. Much of this occurs below conscious awareness. Threat detection operates with similar efficiency. When encountering a potential danger, such as a sudden movement in peripheral vision, neural pathways involving the amygdala can initiate a rapid response before detailed cortical analysis is complete. Heart rate may increase, muscles may tense, and attention may narrow before the stimulus is fully interpreted. This rapid appraisal system prioritizes survival over reflection. Importantly, this does not mean that the brain acts irrationally. It means that multiple processing layers operate in parallel. Fast systems evaluate immediate relevance, while slower systems analyze context and long-term consequence. Awareness often arises after the initial appraisal has already influenced physiology. The sense of agency, the feeling that I decided, remains powerful. It organizes memory and responsibility. Yet the brain's architecture suggests that agency is constructed from layered processes. Signals accumulate, patterns reach threshold, and action emerges. Consciousness observes, integrates, and sometimes intervenes, but it is not always the originator. The illusion is not that we lack control entirely. It is that control is simpler than it truly is. Decision-making resembles a committee rather than a single voice. Some members speak early and quietly, shaping direction before formal approval. Awareness enters the discussion at a later stage. This layered structure explains why habits can persist even when consciously rejected. Repeated behaviors strengthen neural pathways that operate with minimal oversight. Changing a habit requires sustained attention precisely because automatic systems are efficient and deeply embedded. The same architecture enables rapid social judgment. Within moments of meeting someone, impressions form based on posture, tone, and expression. These impressions may later be revised through reflection, but their initial formation occurs automatically. The brain synthesizes fragments of information into a coherent assessment before conscious reasoning begins. None of this eliminates responsibility or choice. It reframes them. Choice emerges from neural processes that extend beyond immediate awareness. The conscious mind participates, evaluates, and can redirect. But it is part of a larger system that has already begun processing. When you decide to move your hand, greet a stranger, or step away from a perceived threat, the decision feels unified. Beneath that unity lies a sequence: preparatory signals, rapid evaluations, pattern matching, physiological adjustment, and finally, awareness of intention. The brain does not wait for permission to begin working. It anticipates, prepares, and often acts before the narrative of choice catches up. Control remains real, but it is distributed, layered, and temporally extended. In this quiet timing gap between neural initiation and conscious recognition, the brain reveals something essential about itself. It is not a passive instrument awaiting command. It is an active system, continuously shaping perception and action, even before we know that it has begun. The brain does not passively wait for the world to arrive. It anticipates. Long before a sound is fully heard or a movement is completed, neural systems are already generating expectations about what will happen next. Perception, in this view, is not a simple recording of reality. It is a negotiation between incoming signals and prior prediction. This idea is formalized in what is known as predictive coding theory. Predictive coding proposes that the brain constantly builds internal models of the world and compares incoming sensory information against those models. When reality matches expectation, processing remains efficient. When there is a mismatch, the brain updates its model. Perception becomes a cycle of prediction and correction. Neuroscientist Carl Friston expanded this idea into a broader framework sometimes referred to as the free energy principle. In simplified terms, the model suggests that biological systems attempt to minimize surprise, meaning they continuously adjust internal predictions to reduce the difference between expectation and experience. The brain, therefore, is not simply reacting. It is minimizing error. This predictive structure explains why perception feels stable even in noisy environments. When you walk through a familiar room in dim light, you do not consciously calculate each object's position. Your brain predicts the layout based on prior experience. Sensory input confirms or slightly adjusts the prediction. Stability arises from anticipation. Prediction also extends into the immediate future. Before you catch a ball, your motor system estimates its trajectory. Before someone finishes a sentence, your language system anticipates the final word. These anticipations are not deliberate guesses. They are automatic simulations running beneath awareness. The brain generates forward models, meaning internal projections of likely outcomes. Sleep offers another setting where prediction becomes vivid. During dreaming, particularly in rapid eye movement sleep, the brain constructs immersive scenarios without external input. These scenarios are not random fragments. They often follow narrative structure with spatial continuity and emotional coherence. The dreaming brain simulates environments, characters, and events, drawing on stored memory fragments to create plausible worlds. Some researchers propose that dreaming functions partly as a simulation platform, allowing the brain to rehearse responses to social or environmental challenges. While this hypothesis remains debated, it aligns with the broader principle that the brain is oriented toward forecasting. Even in isolation from sensory input, it generates models. Déjà vu provides a subtle example of predictive processing gone slightly out of alignment. Déjà vu refers to the sensation that a present moment feels familiar despite the recognition that it is new. One proposed explanation suggests a brief mismatch between memory circuits and perceptual input, creating the illusion of prior experience. The brain's familiarity systems may activate inappropriately, signaling recognition before contextual verification occurs. The result is a fleeting but convincing impression of repetition. Expectation also shapes bodily experience. The placebo effect illustrates how predictive systems influence physiology. When a person believes a treatment will reduce pain, neural circuits involved in pain modulation can become active even if the treatment contains no pharmacologically active substance. Brain imaging has shown that expectancy can alter activity in regions associated with pain perception. The predicted relief becomes partially realized through endogenous neurochemical pathways. Prediction extends beyond physical sensation into social interaction. When entering a conversation, the brain generates expectations about tone, reaction, and likely responses. Subtle cues such as facial expression and posture are integrated into rapid simulations of possible outcomes. Social fluency depends on this anticipatory modeling. Without it, interaction would feel disjointed. The predictive brain does not guarantee accuracy. It prioritizes efficiency. When predictions are strong, they can override weak or ambiguous sensory signals. This can lead to misperception. Optical illusions exploit this principle by presenting stimuli that trigger familiar patterns, leading the brain to fill in details that are not objectively present. Perception becomes an informed guess. Importantly, prediction does not eliminate learning. On the contrary, error drives adaptation. When outcomes differ from expectation, prediction errors arise. These discrepancies prompt neural updating. In this sense, surprise becomes instructive. The brain refines its internal model to better match the environment. Across perception, dreaming, memory anomalies, placebo responses, and social interaction, a consistent pattern emerges. The brain is less a recorder of reality and more a generator of hypotheses about it. It projects forward, tests, and adjusts. Awareness receives the result of this ongoing computation. The world feels immediate and continuous, yet beneath that continuity lies simulation. Each moment is shaped not only by what arrives through the senses, but by what the brain expects to arrive. The present is partly constructed from the future the brain anticipates. In this quiet predictive rhythm, experience becomes an active process. The brain does not wait to be informed. It prepares, proposes, and revises. And through that cycle of simulation and correction, perception remains stable enough to navigate a changing world. One of the most persistent claims is the idea that humans use only 10% of their brain. The statement appears in advertisements, motivational speeches, and films, usually implying vast unused potential waiting to be unlocked. Neuroscience does not support this claim. Brain imaging studies consistently show activity distributed across nearly all regions, even during simple tasks. While not all neurons fire simultaneously, different areas contribute to different functions. And over the course of a day, virtually the entire brain is engaged in some capacity. The origin of the 10% myth is unclear. It may have emerged from early misunderstandings of neurological research, or from misinterpretations of statements about cognitive reserve. Whatever its source, modern evidence contradicts it. Another popular simplification divides people into left-brained and right-brained types, suggesting that analytical thinkers rely predominantly on the left hemisphere, while creative individuals depend on the right. While it is true that certain functions show lateralization, meaning they are more dominant in one hemisphere than the other, this does not translate into personality categories. Language processing often involves left hemisphere regions, and certain aspects of spatial processing may rely more heavily on the right. However, complex tasks require coordination across both hemispheres through structures such as the corpus callosum. Brain imaging consistently demonstrates bilateral activation during creative and analytical tasks alike. Creativity involves networks spanning frontal, parietal, and temporal regions across both sides of the brain. Analytical reasoning similarly engages distributed circuits. The notion that individuals are fundamentally left or right-brained oversimplifies a deeply interconnected system. The so-called Mozart effect provides another example of a finding that grew beyond its evidence. In the 1990s, a small study reported that participants who listened to a Mozart sonata showed a temporary improvement on a specific spatial reasoning task. The effect was modest and short-lived, lasting only minutes. Popular media, however, transformed the finding into a broader claim that listening to classical music increases intelligence. Subsequent research failed to replicate strong, lasting cognitive gains from passive music listening alone. Music can influence mood and arousal, and these changes can temporarily affect performance. But there is no robust evidence that listening to Mozart permanently increases IQ. The appeal of the idea may lie in its simplicity. Intelligence, enhanced by exposure rather than effort. Science tends to be less accommodating. Commercial brain training programs have also promised broad cognitive enhancement through targeted exercises. These applications often involve memory games, pattern recognition tasks, or speed challenges. While practice can improve performance on the specific tasks trained, the evidence for generalized improvement across unrelated cognitive domains remains limited. Transfer effects, meaning improvements that extend beyond the trained activity, are often small or inconsistent. This does not mean mental exercise is useless. Learning new skills, engaging in complex problem solving, and maintaining social interaction are associated with cognitive resilience. However, structured games alone are unlikely to produce dramatic increases in overall intelligence. The brain improves most reliably through sustained, varied engagement rather than narrow repetition. Nootropics, sometimes described as cognitive enhancers or smart drugs, represent another area where enthusiasm can outpace evidence. Some substances, such as caffeine, have well-established effects on alertness. Others, including prescription medications developed for specific neurological conditions, may enhance attention in clinical populations. However, the effectiveness of many over-the-counter supplements marketed for cognitive enhancement remains uncertain. Research into nootropics often reveals modest or context-dependent effects. Some compounds may improve certain aspects of cognition under particular conditions, such as sleep deprivation. Yet claims of dramatic, across-the-board intelligence boosts are not supported by strong, consistent data. The brain operates within biological constraints, and chemical modulation has limits. The idea of photographic memory, the ability to recall images or text with perfect fidelity after brief exposure, also persists in popular imagination. Scientific documentation of true photographic memory in adults is extremely limited. What is sometimes described as eidetic memory, more commonly observed in children, refers to the temporary retention of visual images with high detail for a short duration. Even in these cases, the ability fades quickly and does not function as an exact, permanent recording. Exceptional memory feats observed in memory competitions rely on structured mnemonic strategies rather than innate photographic capacity. Participants use techniques such as spatial encoding and vivid imagery to organize information. Their recall appears effortless, but is built on deliberate method. The brain excels at pattern and association, not literal duplication of sensory input. Why do these myths endure? Part of their appeal lies in empowerment. The idea that 90% of the brain lies dormant suggests untapped brilliance. The belief in hemispheric personality types offers a tidy explanation for differences. Music-enhancing intelligence promises effortless improvement. Smart drugs imply shortcut. Photographic memory suggests hidden potential. Scientific correction does not diminish the brain's complexity. It refines our understanding of it. The brain is powerful, but its power does not reside in secret unused chambers or magical amplification. It resides in dynamic networks shaped by genetics, development, experience, and environment. In place of myth, research offers nuance. Cognitive ability varies across individuals and across contexts. Training effects depend on duration and relevance. Chemical influences operate within physiological bounds. Memory excels at meaning and association rather than perfect capture. Hemispheres collaborate more than they compete. The absence of dramatic shortcuts does not make the brain less remarkable. It makes it more consistent with biology. Improvement requires engagement. Adaptation requires repetition. Understanding requires evidence. When myths fall away, what remains is a system neither underused nor easily transformed by single interventions, but continuously active and responsive. The brain does not hide vast reserves behind arbitrary percentages. It functions as a whole, integrated structure, shaped by experience and constrained by physiology. In this measured clarity, the brain appears not diminished, but grounded. Its real capacities do not require exaggeration. They unfold steadily, within limits that make learning meaningful and growth earned rather than unlocked. The human brain is powerful, adaptable, and efficient, yet it is not limitless. Its performance operates within biological constraints shaped by energy, time, and physiology. These boundaries do not diminish its capabilities. They define them. Understanding real limits allows us to see how the system balances ambition with conservation. Energy is the first boundary. Although the brain represents a small fraction of body weight, it consumes a significant share of available metabolic resources. Neural signaling requires constant maintenance of electrical gradients across cell membranes, and this process depends heavily on glucose and oxygen. When blood sugar drops too low or oxygen supply is interrupted, cognitive function declines rapidly. The brain cannot store large energy reserves. It depends on continuous supply. This metabolic demand explains why prolonged intense focus can feel tiring. While the total energy consumption of the brain remains relatively stable, specific networks increase their activity during demanding tasks. Over time, maintaining sustained activation places pressure on regulatory systems. Fatigue is not simply psychological reluctance. It reflects resource management. Sleep provides another essential constraint. During sleep, especially deep non-rapid eye movement stages, the brain engages in processes that restore balance. One of these processes involves the glymphatic system, a network that clears metabolic waste from neural tissue. When sleep is reduced or fragmented, this clearance becomes less efficient. The result is not only subjective tiredness, but measurable declines in attention, memory, and emotional regulation. Even a single night of sleep deprivation can impair reaction time and decision-making. Chronic sleep restriction has cumulative effects. Functional imaging studies show reduced activity in the prefrontal cortex after insufficient sleep. This region is associated with planning, impulse control, and working memory. As sleep loss accumulates, errors increase and emotional reactivity becomes more pronounced. Decision-making itself is subject to limits. The concept of decision fatigue refers to the gradual deterioration in decision quality after prolonged periods of choice-making. While the underlying mechanisms remain debated, research suggests that repeated cognitive control tasks can temporarily reduce self-regulation capacity. The brain's executive systems are not infinite in stamina. They benefit from intervals of rest. Multitasking presents another commonly misunderstood boundary. The brain does not truly perform multiple demanding tasks simultaneously. Instead, it switches rapidly between them. This switching incurs a cost. Each transition requires reorienting attention and reactivating relevant neural networks. Performance accuracy and speed often decline as task complexity increases. Divided attention fragments efficiency. Attention itself operates within narrow bandwidth. Working memory, meaning the capacity to hold and manipulate information over short periods, is limited to a small number of items at once. Classic estimates suggest around 7 items, though more recent research proposes closer to 4. When information exceeds this capacity, errors increase. The brain must compress or discard input to maintain coherence. Modern digital environments challenge these limits. Constant notifications, rapid content shifts, and frequent task switching engage attention repeatedly. Over time, this pattern may encourage shallow processing rather than sustained focus. The brain adapts to patterns of stimulation. When interruptions become frequent, maintaining deep concentration becomes more difficult. Stress introduces additional constraints. Acute stress activates the release of cortisol and other stress hormones. In short bursts, this response enhances vigilance and mobilizes energy. However, chronic stress can impair prefrontal cortex function while strengthening activity in more reactive regions such as the amygdala. Under prolonged stress, impulse control weakens and emotional reactivity intensifies. The balance between reflective and automatic processing shifts. This stress-related modulation affects memory as well. High cortisol levels over extended periods have been associated with structural changes in hippocampal regions involved in memory consolidation. The brain's adaptability means it responds to persistent environmental signals. When stress becomes constant, neural priorities adjust accordingly. Aging represents another natural boundary. Across the lifespan, certain cognitive abilities show gradual change. Processing speed often slows, and working memory capacity may decrease modestly. These changes reflect shifts in neural efficiency, myelination, and synaptic density. However, not all functions decline uniformly. vocabulary, and accumulated knowledge, sometimes referred to as crystallized intelligence, often remain stable or even increase. Neuroplasticity continues into older adulthood, though its pace may differ from earlier years. Learning remains possible, but repetition and context may play a larger role. Structural imaging reveals that while some regions show volume reduction with age, others maintain stability. The brain ages heterogeneously rather than uniformly. Importantly, limits do not equate to fragility. Boundaries provide structure. Energy constraints encourage efficiency. Sleep cycles regulate restoration. Attention limits prioritize relevance. Stress responses prepare for challenge, even if prolonged exposure becomes costly. Aging introduces adaptation rather than abrupt failure. Recognizing these constraints does not diminish the brain's sophistication. It reveals how precisely calibrated it is, operating within a metabolic budget, a circadian rhythm, and changing environmental demands. Its performance shifts as energy, attention, and stress levels fluctuate. When its boundaries are respected, stability follows. When they are repeatedly exceeded, strain becomes visible. It is a biological system shaped by trade-offs, achieving remarkable function because it works within the limits that sustain it. The future of brain science is not unfolding in abstraction. It is unfolding in laboratories, hospitals, and engineering centers where biology meets circuitry. For much of history, the brain could only be studied indirectly, through behavior or after death. Today, living neural activity can be observed, recorded, and in some cases influenced in real time. The boundary between observation and intervention has begun to narrow. One of the most influential tools in modern neuroscience is functional magnetic resonance imaging, commonly known as fMRI. This technology does not measure thoughts directly. Instead, it detects changes in blood oxygenation, known as the BOLD signal, meaning blood oxygen level dependent contrast. When a region of the brain becomes more active, it consumes more oxygen and local blood flow increases. fMRI captures these subtle changes, allowing researchers to map patterns of activity associated with memory, emotion, language, and decision-making. fMRI has transformed research by making the living brain visible in action. Scientists can observe which networks engage during problem solving, how pain is processed, or how attention shifts across tasks. The images are often displayed in bright colors, layered over grayscale anatomical scans. Yet behind the visuals lies statistical modeling and careful interpretation. fMRI does not read thoughts. It identifies correlations between neural activity and behavior. Beyond imaging, another frontier involves direct interaction with neural signals. Brain-computer interfaces, often abbreviated as BCIs, are systems that translate neural activity into external commands. These interfaces typically rely on electrodes that detect electrical signals generated by populations of neurons. By decoding these patterns, computers can interpret intended movement or communication. Early applications of BC is focused on restoring function for individuals with paralysis. In controlled experiments, participants have learned to move robotic arms or control computer cursors using neural signals alone. The process requires calibration and training. Electrodes capture activity from motor regions. Algorithms interpret patterns, and external devices respond accordingly. The system functions as a bridge between intention and action when normal motor pathways are disrupted. One high-profile company working in this area is Neuralink, founded by Elon Musk. Neuralink aims to develop implantable devices capable of recording and stimulating neural activity with high precision. The company has demonstrated prototypes in animal models and received approval for limited human trials. The stated goals include assisting individuals with neurological disorders and eventually expanding communication between brains and digital systems. While the long-term vision remains ambitious, current efforts focus on clinical applications. Memory research has also moved toward intervention. Scientists investigating memory prostheses are exploring ways to support or restore memory function in individuals with hippocampal damage. In experimental settings, researchers have recorded neural activity patterns associated with successful memory encoding and attempted to replicate or stimulate similar patterns artificially. Early trials suggest that targeted stimulation may enhance performance on certain memory tasks. The field remains experimental, but the concept reflects a shift from observing memory to attempting to assist it. Epilepsy treatment provides another example of technology integrated with neural function. For individuals with drug-resistant epilepsy, implantable devices can monitor brain activity and deliver electrical stimulation when abnormal patterns are detected. Such implants illustrate a broader principle. Neural activity is electrical. Electrical systems can sometimes be modulated. Deep brain stimulation, used in conditions such as Parkinson's disease, applies controlled stimulation to specific regions to alleviate motor symptoms. The mechanism involves altering abnormal neural firing patterns. While precise pathways remain under investigation, clinical outcomes demonstrate that targeted modulation can influence behavior and movement. In parallel with clinical applications, government research agencies have invested in advanced neurotechnology. The Defense Advanced Research Projects Agency, known as DARPA, has funded projects aimed at improving neural interface precision, restoring sensory function, and developing adaptive prosthetic control systems. Despite rapid progress, significant challenges remain. Neural signals are complex and variable. Electrodes can degrade over time. Immune responses may affect implant stability. Decoding algorithms must distinguish meaningful patterns from noise. Safety, longevity, and ethical oversight are central considerations. The brain is not a standardized machine. It is a living tissue shaped by individual variability. Importantly, current technologies do not upload memories or merge minds with artificial intelligence in the way popular fiction sometimes suggests. They operate within defined parameters. Brain-computer interfaces decode limited types of signals. Imaging tools infer patterns rather than extract content. Memory prostheses assist specific functions under experimental conditions. The gap between scientific capability and imaginative speculation remains substantial. As technology advances, questions naturally follow. Who will have access to enhancement tools if they expand beyond medical necessity? How should data derived from neural recordings be protected? What constitutes consent when devices interface directly with cognitive processes? These questions are not secondary. They accompany each technical breakthrough. The future of brain science is neither utopian nor dystopian. It is incremental. Each device, each imaging technique, each implant reflects years of testing and refinement. The brain remains complex, resistant to simplification. Technology approaches it with increasing sophistication, but also with restraint. In this meeting of circuits and cells, the goal is not to replace biology, but to understand and assist it. Observation has become interaction. Interaction has become modulation. And the boundary between mind and machine, once purely theoretical, now rests in laboratories, where electrodes record faint electrical whispers from living neurons. The future does not erase the brain's mysteries. It approaches them carefully, instrument by instrument. Even as technology advances and neural signals become measurable, one question remains quietly unresolved. What is consciousness? Not in poetic terms, but in biological ones. What transforms electrical activity into lived experience, into the feeling of being aware? Neuroscience has proposed several frameworks to approach this question. One influential model is integrated information theory, developed by Giulio Tononi. This theory suggests that consciousness corresponds to the amount of integrated information generated by a system. In simplified terms, It proposes that experience arises when information is not only processed, but unified into a coherent whole that cannot be reduced to independent parts. Integrated information theory attempts to quantify consciousness through a value known as phi, a measure of how interconnected and irreducible a system's internal states are. According to this view, a highly integrated network produces richer experience than a fragmented one. The theory does not claim to solve subjective awareness entirely, but it offers a structured way to think about it mathematically. Another influential framework is global workspace theory, introduced by Bernard Bars. This model compares consciousness to a stage illuminated by a spotlight. Many processes occur backstage in parallel, but only a small portion becomes globally available, entering a workspace where information can be broadcast to multiple systems such as memory, language, and decision-making networks. In this view, consciousness arises when information becomes widely accessible across the brain. While these theories differ in emphasis, both attempt to move the discussion from philosophy toward measurable criteria. Researchers have begun exploring ways to estimate levels of consciousness in patients with brain injury by analyzing neural complexity and connectivity. Techniques involving perturbational complexity index, which measures how the brain responds to controlled stimulation, offer one approach to assessing conscious capacity without relying solely on behavioral response. Yet measuring consciousness remains challenging because it is inherently first person. Brain imaging can reveal patterns of activation, but it cannot directly capture subjective experience. It can indicate correlates, not the feeling itself. The distinction between neural activity and lived awareness remains a central puzzle. Comparisons with artificial intelligence add another dimension. Modern AI systems can process language, recognize images, and generate responses that resemble human reasoning. However, current AI operates through pattern recognition and statistical association. There is no evidence that it possesses subjective awareness. The difference between simulating intelligence and experiencing consciousness is significant, though difficult to define precisely. This comparison raises deeper questions. If consciousness depends on integration and information exchange, could sufficiently complex machines develop something analogous? Or is biological substrate essential? Neuroscience has not reached consensus. The question remains open, framed cautiously within empirical boundaries. The discussion extends beyond humans. Studies of animal cognition reveal sophisticated behaviors in species such as primates, dolphins, corvids, and octopuses. Mirror self-recognition, problem-solving, and social learning suggest varying degrees of awareness. Determining where consciousness begins and how it varies across species challenges simple categories. The boundary is not sharp. Ethical implications naturally follow. If certain animals possess richer conscious experience than previously assumed, standards for treatment may require revision. If technology allows deeper interaction with neural systems, consent and privacy become critical. The brain is not merely an organ. It houses identity, memory, and agency. Intervening in its processes carries moral weight. The unanswered question persists not because science lacks effort, but because the phenomenon itself is subtle. Consciousness does not announce its mechanism. It is the background condition for every observation. Studying it is unusual because the observer and the subject are the same system. The brain is examining itself. Neural circuits generate theories about neural circuits. Imaging devices designed by human cognition are used to interpret human cognition. The investigation folds inward. This recursive quality adds both depth and limitation. As theories evolve and data accumulates, clarity increases incrementally. Certain neural patterns correlate reliably with conscious states. Connectivity appears crucial. Complexity matters. Yet the transition from electrical signaling to felt experience remains partly obscured. Perhaps the most careful conclusion is this: consciousness is neither mystical nor fully mapped. It is grounded in biology, shaped by networks, and constrained by physiology. At the same time, its subjective dimension resists complete reduction. In the quiet intersection of philosophy, neuroscience, and technology, the question remains open. The brain continues its activity, generating awareness that studies itself, refining models, adjusting hypotheses. The search does not diminish the mystery. It refines it, slowly, with patience rather than spectacle.